Innate immunity, inflammation, and wound healing. The human immune system protects the body through a series of carefully coordinated defenses. The first line of defense is called innate immunity and includes physical, mechanical, and biochemical barriers that block the entry of harmful microorganisms. If these barriers are breached, the second line of defense is triggered, which is inflammation. This response acts quickly to contain and eliminate threats. If further support is needed, the body activates the third line of defense, adaptive or acquired immunity, which targets specific pathogens with a highly specialized response. The first line of defense relies heavily on physical barriers. The skin serves as a protective shield, continuously sloughing off dead cells along with any attached microbes. The linings of the gastrointestinal, genitourinary, and respiratory tracts also provide critical protection. Mucus and tiny hair-like structures called cilia trap microorganisms, which are then expelled from the body through actions like coughing, sneezing, urination, vomiting, or defecation. In addition to physical barriers, the body produces chemical defenses derived from cells. Substances like saliva, tears, earwax, sweat, and mucus contain antimicrobial components. Lysozymes found in these secretions attack bacteria by breaking down their cell walls. Specialized peptides such as defensins and collectins directly kill bacteria, fungi, and viruses, strengthening the body's first line of defense. Another crucial component of innate immunity is the normal microbiome. Every surface of the human body is colonized by a unique collection of bacteria and fungi that live in harmony with us. These microorganisms produce enzymes that aid digestion, synthesize important metabolites, release substances that prevent pathogen growth, and compete with harmful microbes for nutrients. They also foster the development of the adaptive immune system and may even help regulate communication between the brain and the gastrointestinal tract. When pathogens breach physical barriers, the inflammatory response is activated. Inflammation occurs in vascularized tissues and acts quickly, within minutes. It involves a complex interaction between cellular and chemical components to contain the injury, eliminate harmful agents, and begin the healing process. Unlike the adaptive immune response, inflammation is nonspecific, meaning it reacts the same way to many different threats. The vascular response to inflammation is essential for delivering immune cells to the site of injury. When inflammation begins, hemostasis, or coagulation, prevents excessive bleeding. Blood vessels then dilate, allowing more blood to flow to the affected area. Increased vascular permeability enables white blood cells, fluids, and proteins to leave the circulation and enter the tissue. White blood cells adhere to the inner walls of the vessels and migrate through them to reach the site of damage. Locally, this process results in the familiar signs of redness, heat, swelling, pain, and loss of function. Inflammation serves several protective purposes. It helps prevent and limit infection and further tissue damage. It controls the inflammatory process itself to prevent excessive harm. Inflammation also prepares the injured area for healing and repair. Importantly, it creates conditions that facilitate the development of an adaptive immune response, ensuring the body is better prepared for future attacks. Three major plasma protein systems support the inflammatory response: the complement system, the clotting system, and the kinin system. These systems consist of proteins and enzymes circulating in the blood. They are activated in a tightly regulated sequence called a cascade, where each step triggers the next to amplify the response. The complement system is one of the body's most potent defenses. It produces biologically active fragments that enhance inflammation and destroy pathogens. Activation of key proteins like C3 and C5 generates molecules such as opsonins, which tag pathogens for destruction. chemotactic factors, which attract immune cells, and anaphylatoxins, which promote inflammatory reactions. The complement system can be activated through three different pathways: the classical, the lectin, and the alternative pathways. The clotting system forms a fibrinous meshwork at the site of injury or inflammation. This mesh traps pathogens, prevents the spread of infection, and forms a clot to stop bleeding. It also creates a supportive framework for tissue repair and healing. The clotting system can be initiated through two pathways: the tissue factor pathway, also known as the extrinsic pathway, and the contact activation pathway, known as the intrinsic pathway. The kinin system plays an important role in amplifying inflammation. Its primary molecule, bradykinin, causes dilation of blood vessels, promotes pain, triggers smooth muscle contraction, and increases vascular permeability. By interacting closely with the clotting system, the kinin system helps coordinate the body's inflammatory defense. Because these plasma protein systems are so powerful, they must be tightly controlled to avoid excessive damage. Multiple regulatory mechanisms are built into the system to ensure that inflammation is effective, but not overwhelming. Proper regulation allows the immune system to clear threats while preserving healthy tissue. When tissues are injured, various cellular components are activated. These cells respond to molecular signals at the site of damage and are recruited to help contain and repair the injury. Through surface receptors, these cells recognize danger signals, initiate intracellular signaling pathways, and perform critical functions. They confine the extent of damage, kill microorganisms, remove cellular debris, and activate healing processes. Cells involved in inflammation use pattern recognition receptors, or PRRs, to monitor for signs of danger. These receptors recognize two types of molecular patterns: pathogen-associated molecular patterns, known as PAMPs, and damage-associated molecular patterns, called DAMPs. Different types of PRRs include toll-like receptors, C-type lectin receptors, NOD-like receptors, complement receptors, and scavenger receptors. Together, they allow immune cells to rapidly detect and respond to threats. Cytokines are critical signaling molecules that activate immune cells and regulate the inflammatory response. Chemokines, a specific subset of cytokines, act as chemical attractants. They draw leukocytes, or white blood cells, to sites of injury and infection. Chemokines are produced by a wide variety of cells, including macrophages, fibroblasts, and endothelial cells. More than 50 different chemokines have been identified, each helping guide the immune response. Interleukins, or ILs, are a major category of cytokines produced primarily by macrophages and lymphocytes. They are synthesized in response to activation of pattern recognition receptors or stimulation by other cytokines. Interleukins regulate the behavior of immune cells by altering cell adhesion molecules, attracting leukocytes to areas of infection or injury, promoting the growth and maturation of immune cells, and enhancing or suppressing both inflammation and the adaptive immune response. Several cytokines have strong pro-inflammatory effects. Tumor necrosis factor alpha, known as TNF-α, is one of the most potent. It induces fever, increases the production of inflammatory proteins in the bloodstream, and can cause muscle wasting and blood clot formation within vessels. Interleukin-1, or IL-1, also enhances innate and adaptive immunity and acts as a growth factor for immune cells. Interleukin-6, or IL-6, stimulates liver cells to produce inflammatory proteins, and when present in very high amounts can be lethal. In contrast, some cytokines have anti-inflammatory effects. Interleukin-10, or IL-10, suppresses the activation and proliferation of immune cells, helping to limit excessive inflammation. Transforming growth factor beta, or TGF-beta, also suppresses the immune response by downregulating the production of pro-inflammatory cytokines. Like IL-6, very high levels of these molecules can be harmful, highlighting the need for careful immune regulation. Interferons are cytokines specialized in fighting viral infections. Type I interferons are produced and released by virally infected host cells. They stimulate the production of antiviral proteins in neighboring healthy cells, helping prevent the spread of the virus. Type II interferons, primarily produced by lymphocytes, activate macrophages and enhance their ability to detect and destroy abnormal cells. Mast cells are powerful activators of the inflammatory response. They are packed with granules containing a variety of chemical mediators. These mediators are released in response to tissue injury in two ways: degranulation, which is the immediate release of pre-stored granules, and synthesis, where new mediators are produced and released in response to ongoing stimulation. Degranulation is a rapid process that occurs within seconds of mast cell activation. It results in the release of stored biochemical mediators into the surrounding tissue, leading to an immediate and robust inflammatory response. During degranulation, mast cells release critical molecules like histamine, chemotactic factors, and cytokines. These substances contribute to the hallmark signs of inflammation, such as swelling, redness, and increased blood flow. Histamine, one of the primary mediators, is a vasoactive amine. It causes the temporary and rapid constriction of smooth muscles, followed by the dilation of small blood vessels, especially in postcapillary venules. This increases blood flow and vascular permeability. Histamine acts through two main receptors: the H1 receptor, which promotes inflammation, and the H2 receptor, which has anti-inflammatory effects. Chemotactic factors released by mast cells create chemical gradients that attract specific immune cells to the site of injury. Neutrophil chemotactic factors guide neutrophils, while eosinophil chemotactic factors specifically attract eosinophils, playing a crucial role in orchestrating the immune response. In addition to releasing stored mediators, mast cells also synthesize new inflammatory mediators. Leukotrienes have similar effects to histamine, but act later in the inflammatory response. Prostaglandins, which are also produced, help intensify inflammation and trigger pain. Drugs like aspirin and other nonsteroidal anti-inflammatory drugs block the production of prostaglandins, reducing both inflammation and pain. Platelet activating factor has effects similar to leukotrienes and also plays a role in activating platelets. Endothelial cells line the interior surface of blood vessels and play a critical role in regulating circulation through the microvasculature. They help control the movement of water and solutes between the blood and surrounding tissues. When endothelial cells are damaged, it triggers a series of responses. Platelets are activated, leukocytes are recruited to the site, and the healing process is initiated. Healthy endothelial cells are essential for maintaining balance between inflammation, blood flow, and tissue repair. Platelets are small, cell-like structures activated by vascular injury. Once activated, they interact with the clotting system to help stop bleeding. In addition to forming clots, platelets release granules filled with signaling molecules. One of the substances released is serotonin, which functions similarly to histamine, promoting vasodilation and increasing vascular permeability. Platelets therefore serve both as responders to bleeding and contributors to inflammation. Neutrophils, also known as polymorphonuclear neutrophils or PMNs, are the most abundant type of white blood cell and the primary phagocytes involved in early inflammation. These cells rapidly migrate to sites of infection or injury, where they ingest bacteria, dead cells, and cellular debris. Neutrophils are short-lived and eventually die at the site of inflammation, becoming part of the purulent, or pus-like, exudate that forms as the immune system cleans up the damage. Eosinophils are mildly phagocytic cells that play a specialized role in defending the body against parasites. They also help regulate the effects of other inflammatory mediators, preventing an overreaction of the immune response. Eosinophils are especially important in allergic reactions and parasitic infections, where they contribute to tissue defense and inflammation control. Basophils are the least common type of granulocyte, but have a significant impact on immune responses. Their granules contain substances similar to those found in mast cells, including histamine and heparin. Basophils are important sources of cytokines, which help direct immune cell behavior. Despite their low numbers, basophils are key players in allergic reactions and in supporting inflammation. Monocytes are produced in the bone marrow and circulate in the blood. When they are drawn to an inflammatory site, they exit the bloodstream and transform into macrophages. Unlike neutrophils, monocyte-derived macrophages arrive later, typically after 24 hours, but they live longer and play a vital role in cleaning up debris and supporting tissue repair. These macrophages are highly effective at engulfing pathogens and releasing growth factors for healing. Dendritic cells are located in peripheral tissues, such as the skin and mucosal surfaces. When they detect pathogens, they capture antigens and migrate through the lymphatic system to lymph nodes. There, they present these antigens to T lymphocytes, initiating an adaptive immune response. Dendritic cells serve as a critical link between innate and adaptive immunity, ensuring that the body develops long-term protection against specific pathogens. Lymphocytes include T cells and B cells, which are central to adaptive immunity. They also support the activity of macrophages and help coordinate the immune response. Natural killer cells are a distinct type of lymphocyte that specialize in eliminating virus-infected and cancerous cells. These cells use inhibitory and activating receptors to distinguish between healthy and abnormal cells. In addition, they release cytokines and toxic molecules that directly kill their targets. Phagocytosis is the process by which immune cells such as neutrophils and macrophages ingest and destroy microbes, particles, or dead cells. This process begins with the production of adhesion molecules that allow phagocytes to stick to the inner walls of blood vessels. The cells then undergo margination, where they align along the vessel wall, and diapedesis, where they move through the vessel wall into the surrounding tissue to reach the site of infection. Once at the site of injury, phagocytes recognize their targets through receptors that bind to pathogens or damaged cells. These interactions prepare the immune cell for ingestion, a key step in clearing the area of threats and debris. Phagocytosis occurs in 4 key steps. First, the immune cell recognizes and adheres to the target. Second, it engulfs the particle, forming a membrane-bound vesicle called a phagosome. Third, the phagosome fuses with granules or lysosomes, forming a phagolysosome. Finally, destructive enzymes within the phagolysosome break down and eliminate the target. This is one of the most important functions of the innate immune system. Inflammation can be classified as either acute or chronic. Acute inflammation is a rapid, self-limiting response that typically lasts between 8 to 10 days. It is designed to eliminate the cause of injury, clear out damaged cells, and begin tissue repair. However, if the acute response is not successful or the body is continually exposed to harmful stimuli, inflammation may become chronic. Chronic inflammation can persist for weeks or even months and is often associated with tissue damage and long-term health problems. The local manifestations of acute inflammation stem from changes in blood vessels, and the release of inflammatory components into tissues. These changes cause classic signs such as heat, redness, swelling, pain, and loss of function. Additionally, different types of exudate or fluid may be produced at the site. Serous exudate is watery and clear. Fibrinous exudate is thick and sticky with clotting proteins. Purulent exudate, or pus, contains dead white blood cells and debris. Hemorrhagic exudate includes blood due to severe injury. In addition to local effects, acute inflammation also has systemic manifestations. Fever is a common response, caused by pyrogens that reset the body's thermostat in the hypothalamus. Leukocytosis, or an increase in circulating white blood cells, occurs as the body ramps up its immune defense. The liver also increases production of acute phase reactants, plasma proteins that support inflammation. These systemic changes reflect the body's effort to fight infection and restore balance. Chronic inflammation develops when the acute response fails to eliminate the threat or when certain pathogens or irritants resist destruction. It is characterized by ongoing tissue injury, persistent pus formation, and incomplete healing. In some cases, chronic inflammation is caused by microorganisms that can survive inside cells, or by toxins that continuously damage tissue. This prolonged response disrupts normal tissue function and can contribute to disease progression. Several features distinguish chronic inflammation. There is a dense infiltration of immune cells, especially lymphocytes and macrophages. Granulomas may form when the immune system tries to wall off substances it cannot eliminate. Inside these granulomas, specialized cells called epithelioid cells and multinucleated giant cells form. These structures are a hallmark of long-standing inflammation and are often seen in diseases such as tuberculosis. Wound healing involves two main outcomes: regeneration and repair. Regeneration occurs when injured tissues are fully restored to their original structure and function. Repair, on the other hand, replaces damaged tissue with scar tissue. Scar tissue is primarily made of collagen, a strong fibrous protein. While it restores structural integrity, scar tissue lacks the specialized functions of the original tissue. Wound healing can proceed through either primary or secondary intention. Primary intention refers to wounds that close with minimal tissue loss, such as surgical incisions. These wounds heal quickly and with minimal scarring. Secondary intention applies to wounds with significant tissue damage, such as open wounds. These require more time and more tissue replacement, often resulting in larger scars. Wound healing occurs in distinct phases. Phase 1 is hemostasis, or coagulation, which begins immediately after injury. Tissue damage leads to bleeding into the wound, clot formation starts, and platelets become activated. These platelets degranulate and release substances that increase capillary permeability and stimulate the release of growth factors, initiating the healing process. The second phase is inflammation. During this stage, immune cells rush to the wound. Macrophages and mast cells release vasoactive cytokines that widen blood vessels and attract more immune cells. Neutrophils are among the first responders, clearing debris and pathogens. Lymphocytes also arrive to initiate the adaptive immune response. This phase is critical for preparing the wound bed for tissue regrowth. Phase 3 is known as proliferation and new tissue formation. During this stage, the wound is sealed, and the initial clot is gradually replaced with either normal tissue or scar tissue. Macrophages continue to clean the wound and release growth factors that recruit fibroblasts. Fibroblasts produce collagen, while angiogenesis, the formation of new blood vessels, restores circulation. This stage also includes granulation and epithelialization, where new skin begins to cover the wound. Phase 4 is the remodeling and maturation phase. During this final stage, the newly formed tissue continues to differentiate and strengthen. Fibroblasts reorganize the collagen fibers in the scar, while unnecessary cells undergo programmed cell death. Over time, the scar tissue becomes more compact and aligned with the natural structure of the surrounding tissue. This process can take weeks to months, depending on the extent of the wound. This final image, taken from a clinical reference text, illustrates the entire sequence of wound healing from coagulation to remodeling. It shows how complex and well-coordinated each step must be in order for the body to successfully repair tissue after injury. Any disruption in these stages can lead to complications. Wound healing can become dysfunctional at any phase. Factors that interfere with healing include poor blood supply, excessive bleeding, or abnormal fibrin accumulation. Several chronic conditions, such as obesity and diabetes, are known to impair healing. Infections, poor nutrition, certain medications, and smoking can also negatively affect the body's ability to repair itself. When these factors are present, the risk of poor outcomes and prolonged recovery increases significantly. Problems during wound healing may involve improper collagen production, leading to abnormal scar formation. Keloids are thick, raised scars that extend beyond the original wound. Hypertrophic scars are also raised but remain within the wound's boundaries. In some cases, a wound may reopen along a surgical suture line. This is called dehiscence and significantly increases the risk of infection. Another issue is contracture, where excessive wound contraction leads to deformity, often restricting movement. This final image, sourced from medical pathology references, highlights various structural complications that may occur when wound healing fails. These visual examples emphasize the importance of proper care and early intervention to avoid long-term functional or cosmetic issues.